Welcome everybody to this training session of the European Nano Safety Cluster Working Group A on education, training and communication jointly and in cooperative manner with the NanoCommons research infrastructure, which we are building. And I would like to welcome everybody now here to this current session on present experimental workflows, how they can be connected to instance or via instance maps to, for example, electronic laboratory notebooks where the data and eventually also protocols are located and where you can interact with your teams quite cooperatively around the globe. We have had a number of training sessions on that. And we will also like to discuss a bit today on generation of standard operating procedures in line with initiatives that are going on in the direction of creating data, so characterization, templates for nanomaterial characterization, as well as for modeling events. So what are we going to do today? We see here, first I give just a very few glimpse on what we have done so far on training events, where you find materials for that, and then give a short overview on what we are going to do today. So we are going to look into what the instance mapping tool can provide. It's powered by 7past9. Thomas Exner is with me today. We also have Ben Bunt sitting next to me. He's going to present a little bit an application of this instance maps, how we are using it for our experimental workflows in the area of human hazard assessment. Then we're also going to talk a bit about metadata provision, which are so this connection of this instant mapping tool to electronic laboratory notebooks. We then deal with standard operating procedures from our perspective of human hazard assessment, but there we would also like to invite the audience for interaction because you are working in different workflows and in different fields and have your domain-specific requirements eventually for biological data provision. So we are then going to keep this quite hands-on, and we are going to show you, and you can also interactively together with us and ask back to us, do some instant map establishments or developments for your specific experimental workflows. Good. So we have had a number of training events already, and I have mentioned electronic laboratory notebooks now for a couple of times already. So we had a couple of trainings on that, and you would be able to find some background material of how this can be established, what this can provide, and how you can use it also in international teams cooperatively. And I would like to highlight on this topic these two laboratory notebook related training events that we had. So first it was a demo on the features that we have powered by our infrastructure NanoCommons that we have established within the past couple of years, and also more kind of interactive getting hands-on session. You would find in the NanoCommons homepage the access links to these previous events that were provided, and there you find also access to recordings and to the slide sets which are deposited at Zenodo. So you should be able to kind of also get quite decent information on how this can be implemented. This also nicely connects, and eventually we can also discuss this today, to the NanoCommons knowledge base, so where you can upload data, including metadata, in order to be able to satisfy also the needs of data fairness. We have had also a quite dedicated webinar on that, and there is also plenty of interesting recording material around, including slide set available where you can get more information about. Good. Very interesting compilation is basically the NanoCommons Nanomaterial User Handbook, which we have also accessible at the homepage, where topics specifically these kind of events and resource materials are collected. So I would like to, or we would like to invite you to go to the NanoCommons knowledge base, click on the handbook if you want, and then you would see certain different topic areas where you find more information and are nicely linked to information resources regarding that. We also collaborate with the Elixir Tests, the training platform, and you would find, of course, also these training events and access to the materials located there. So this is, for example, just a picture how this looks like, and there is of course some background information, and by clicking here you would be guided to the resource materials, for example. One other activity which we have regularly, and it's a matter of discussion whether we are going to have this next in 2 weeks from now, it's a kind of co-creational instant mapping sessions where we together can help building you your instant maps for your experimental workflows. This now looks very complicated, but we hope that after today's session you are pretty much more familiar with what all these colors mean, and we can discuss this later and can cooperatively then work on your specific workflows in these kind of bi-weekly sessions driven basically by the initiative of Thomas Exner, whose picture you see here. Good, then I would like to highlight and advertise the Nano Week of the Nano Safety Cluster, which happens to be in Cyprus. So you see a nice picture here of Limassol. It's a good coincidence for many projects. Nano Safety Cluster and other projects are meeting there. Be there, be with us on the Thursday of this week in between 20th and 24th of June. We are going to have the training day, where we are also going to look into SOPs, into fairness of data, into grouping, into read across and things like that. Safety and sustainability by design concept on display, decision support systems and things like that. So it's going to be a very interesting, very motivating and exciting week there, with lots of interaction, which we have missed for past 2 years, I would say. So just one short glimpse finally for the introduction on these different links for the resource materials that I have highlighted. And with this, I would like to give the word to Thomas for his introduction on the instant maps, how this story started and where we are standing at the moment. Thanks for being here today.
Okay. Thanks a lot. I just had to unmute myself. Thanks also from my side to everyone who joined this meeting. I'm really happy that so many actually joined, yeah, really from around the globe. It's amazing. It's really cool to see so many people here. Thanks, Martin, for the introduction. I can probably directly jump into what we want to do, and the main point is really that we use these tools, but the tool is really also meant to perhaps change a little bit the concept on how data is managed in large projects, but also in your daily life. Yeah, that you don't wait until the final end until you think about data management, but that this is really something you try to do whenever it comes to. Therefore, what we have to want to do today is have these idea of, is there a way that we can organize complex studies, the thing which Martin already showed, yeah, and then visualize it, and in that way being able to present it easier to others, really going through that, and we will have some examples today how we show that. Just for this is the study I'm doing, these are the different parts I need, is that okay? Yeah, you could go to your colleague or your supervisor or something like that to present that in a way, or perhaps in publications. Yeah, the second thing is then really going into the area of, can we use that actually also for data management? Yeah, is there a way that the structure we build is then also guiding a little bit the documentation of the study design, of the protocols, of SOPs, and so on. Now my cat is coming. And finally to the data. Yeah, and therefore what we do today is a little bit these 7 steps. Yeah, it should be very open, and if you have questions, please don't ask them whenever you have them. I'm happy to ask or also be interrupted if you want. Yeah, but I will start with a little bit of a history. Yeah, what are instance map? Because I actually have to admit that this was not my idea, even if I really think that it's a great one. Then we will look at this tool, which we created based on that. And then also in a live session, then we will look at, is that only something for a lab scientist? Can we do more with it? Can we even use it for creating SOPs? And then I will hand over to Martin, who is the expert there. Then going back a little bit into this area of data again, instance maps and FAIR, and then whenever you're still happy to do something, then you can go in. Many of you should have received a passport today, therefore you can go there. You should be able to see more or less everything I present, and we will then also share more on that in that way. Okay, how it all began. Yeah, and again, it is not my idea. Yeah, I have really to thank people from Nixie and or the NIC and NanoPhase who brought me into contact with that, and also the whole NanoCommons way to do that at the end. Yeah, that it started with a way to look at publications and curate data from publications. Therefore, this is the first point, that you look at something which is already a full study and then try to organize your data in a way that you see what is going on. Yeah. Then the second step was that this was adopted by the NanoPhase project, really for their direct data management. Yeah, it was not paper and then data management, but directly in the project. And all that is now also possible to provide the associated data, in the specific data format to the NanoCommons knowledge base. And what we are planning is that in principle also these instance maps will be there as metadata that to the data sets. And this is something we are working on at the moment. What are instances? Yes, this is the first question, and this is from the NanoPhase NIC curation manual. Just taking from there how to define instances. Instances are something when something happened to your material. Yeah, therefore we will soften this criterion a little bit, but just something where you say, okay, this is something different. Yeah, and it might be that you have some reaction building, or you expose it to another environment, something like that. You have some fate mechanisms or whatever. Yes, or even very simple things like dispersion in a solution, or dispersion in a medium. Therefore, all these little things can be instances, but it really depends a little bit on what your study is actually to understand what an instance is. And you can also go finer and broader if you want. The second thing is that then these instances can be filled with life. On one side, you have the materials which are then associated at to this instance at that specific point of time, or point of fate in a way. And then you can also define here the medium. I would call it more environment or matrix or whatever you want to call it, but just as a starting point, we can call it medium, just the surrounding around your material to describe where it is at that state. And then you can add measurements, and many things, some things, and what you see here is that because it was curated from literature, there are many things which are just partly known, or some which are really measured. The age is something which you also have measured it. Okay, but it's a relatively simple experiment. Anyway, but in that way you can continue and build these up, and you see that this is also the reason why these pictures become relatively quickly complex. But I think the instance maps are still a way to cover this complexity. These are the original categories, yeah, that you have the instances, materials, media, supplementary. This is okay, data in a way, and then you have the properties, whatever that is, yeah. But this is relatively limited. It's clear you don't look at really the data behind it, but it's really just trying to organize that in a way. Okay, you can do that for the nano phase, and again, this is relatively complex. I don't want to go into that detail or that examples. I have others later, but just to say, okay, the whole nano phase project was actually trying or doing that, creating these data files. There was this issue that this creation of the maps was relatively time consuming because there was no real tool for that. But in principle, you could use the data files and create these instance maps from them. All the information is in. Yeah, you can go over that. There is already the idea that this is connected to data. Therefore, you have also these ideas of the instances in the data capturing, where you see here that these instances are covered. There is a description what the material, the medium is, but this is becoming relatively quickly very, very complex, because everything, you don't want to have hundred thousands of these spreadsheet tabs for each individual instance. Therefore, you try to squeeze that in as little as possible way, but this also makes then the need that you link that somehow. Yeah, when you go to the protocols, for example, you have to then have some identifiers which shows you, okay, this protocol belongs there. It's a little bit, yeah, complex, or not a little bit complex, but perhaps even a little bit messy when you have to do that. Therefore, we were thinking, can we go from there, improve on that a little bit? First step would be to really make something which is easier to use, yeah, that you can really produce these instance maps so quickly that you do that for all your experiments, not just for very specific ones which you then have to want to demonstrate, because I really think they are very useful to understand what was going on. Why better? Yeah, and again, just a little bit of a summary here. Nick and NanoPhase in principle looked at the studies or past studies. Yeah, the Nick was really on literature data. NanoPhase was on their own data, but still after, in principle, the whole study was done. Yeah, therefore, they just waited until it was done, then they sat down and created these files. I already mentioned that there was no way to build these easily. You could just use PowerPoint more or less, yeah, to do that, or some other editor, but there was not something specific. I also showed you this relatively complex data schema, and which has the advantage that it's really flexible. That is a good thing, but this flexibility also brings complexity. Yeah, and the other point was that in principle, if you are doing these experiments, you have different people doing things, yeah, and therefore it would be nice if it's not just one person who has to do everything, but that you can really work on that in a collaborative way, yeah, and create these instances, but then also the data files in a more collaborative way. And why? Because then you go here to this data lifecycle, yeah, and this is something we already presented, I think, but which is really discussed a lot in one of the papers. I should have cited it, actually. I have the citations on there, I think. Anyway, but the idea is really that you have here these different stages. You start from planning, collection, the analysis, and then the presentation and the reuse success. Yeah, but in principle, we look at these four things. Yeah, that you have these different steps, and each of these steps is actually producing some kind of metadata or data. Yeah, even the planning is already showing you what kind of experiment are needed, the collection you have, or what protocols you want to use, and so on. Yeah, therefore there is a lot of data already produced at this stage, and it would be great if we could just then also collect these at that state. Yeah, and then the only collection here is really then the experiment, more or less, but with an parallel data documentation. And again, it is done by different people. Yeah, we have first the data creator who is doing, but then also the customer who is selecting or specifying the objectives, discuss the study design. That might be different people than the people who are actually doing the research, or even the research, the experiments might be done by different people. Then you have the processing, modeling, analysis, validation. All that is done by a team of people. Quality control is done by a team of people, and therefore this kind of a collaborative way to do that would be also, I think, quite useful. Okay, I jump over these two. To do that, we try to then extend that a little bit. Yeah, and these are the things we have now. The main point is that, and this is still changing. Yeah, this is a first way to do it, but you will see, and then later when Martin starts to discuss, you will also see that probably we will need more categories. Yeah, but the main change is really that this is what you saw already, more or less. Only the color coding has changed a little bit. Yeah, you have also then the difference that you have some different types of properties. Also there, the naming is not perfect, but that you say, okay, I have something which I get from the literature or from the material provider. Yeah, like they have a specific, they give you a size. You don't know because you didn't measure it yourself. You have to trust that. But being able to show this is not something I measured myself or computed myself, but that is something which is coming from somebody else. You could use this one color, and then you could say, okay, this one is in silico, this other one is in vivo, in vitro experiments, just to also be able to separate these two a little bit. Main point here is really this part that we said, okay, in principle, it would be good to also link directly protocols to that whole thing. You know, that it's not just data or the kind of data you measure, but that you really can also manage the whole process there by putting the protocols in as well as then the data in. Another just a little side comment. Yeah, I think we also become, what we are looking at is becoming more and more complex. Yeah, therefore it's a little bit unclear, the picture, but anyway, the idea is that this separation between medium and material might be a little bit arbitrary, and also a little bit too not flexible enough. Yeah, because you might have your material in a specific product matrix, and then you put this product into a biological system or an environmental system, and therefore these instances or the components in these instances might be relatively complex, and they have also some kind of relationship to each other. Yeah, therefore we are thinking a little bit about how could we change that there is a better way to also describe these more complex settings. But again, just a side comment. This is the tool. Yeah, this looks similar to what Martin was showing. Yeah, it's just the user interface, and we will go live in there in a second. But that is just how it looks like for a relatively complex thing. You can then jump in there and see these different things, the instances, then these protocols showing what actually happened from one instance to the other. Here in this case, you just take graphite and oxidized it in this one step. And then you can also have some kind of properties to these description what was done. But that was the first stage of the system. We are now going more into this. Okay, we add data in a much more flexible way to it, and also more. How are these created? And if you want to start, I think for me it was the best way to just take a publication. And I learned reading publications very different to what I did before, because before I read more or less the conclusion and then some results, and perhaps if then something was interesting, I went into the material and methods. But here I'm concentrating, this is only material and methods. This is the only section which is really interesting to you, therefore to build these instance maps. But you can see here that in principle here we have already this one-to-one mapping. You have this synthesis of graphene oxide. This would be exactly here this part, and we could link this part to the to this node, and say, okay, if you click on that, you would get this information. And then you go through that, and then you say, okay, here's a characterization of the oxide, graphene oxide, and there were some size measurements in there. I don't find that directly, but in principle, all these information which I had here, there were a couple of more. All these are just given in these in this publication, and this is exactly how the NIC was actually using that. Going back into literature, looking at papers and trying to build these instance maps. This is, and again, this is the best way to start with something like that. We now take the next step and go really into these very detailed breaking down the experiment. And for me, this was the first, or was really when we saw that, I thought, or that this will become really then even more beneficial if you are now able to separate these things into these blocks and describe these blocks then also different independently. The main point is, first, you can always directly link the data to it. And when I say data, I mean that very, very generally, a protocol is for me also some kind of data. I can also call it information, but in principle, whenever I say data, I more or less mean everything what you find here. Raw data, process data, yes, but also protocols and even more. And then you can link to that, and you can say, okay, I just show here a little bit what this might look like. The one could be just this kind of relatively complex data structure, really, which would be probably more for a computer to read, to build a data model out of that. But you could also then link that to Synode or to any other electronic lab notebook, where you say, okay, these are now describing my protocols. Or I could even go there and just link an Excel sheet to it with the results. Therefore, at the moment, very, very flexible, whatever you want, and we will have show a little bit more. And this is exactly when I move over. If there are any questions, please go in, and I can just wait a little bit, but otherwise I just continue. This is when you log in, you will see more or less the same screen. Everyone who is training, who came to this training, is showing here shared with training. Therefore, this is what you will see. At the moment, I'm the only person who is sharing something, but if anybody starts to create their instances, then they will also see what will happen. You can then go, for example, to this Corona. This is exactly the one which I also presented in the slides. You can then zoom in there, which is quite nice to look at these different things. You have here that I already explained. You have this oxidization of the graphite, and then you have here size distribution, surface chemistry, a lot of analysis of that. And then they went on and put that into a biological system and measured some toxic effects in that. Therefore that is all fine. If you do that with my instance maps, you will see that you cannot do much. You can move them around if you like. If you want to show them a little bit different, that's helpful. Sometimes you just want to rearrange that to see better what you want to focus on, but that is more or less everything you can do. And this is always the case. Only you can work on your own instance map. If you want to do something with my instance map, you have to copy them, and then I don't share them with anybody. But now this is my one. Therefore, and my one is because I have 2 user accounts. To show how it actually would look like for you. But now I'm the other Thomas, and now I can change anything here. I can add instances by just drag and drop. I can build connections to whatever I want, and I can also then remove them again. I can delete this thing. No, I don't want to delete the whole thing. I first wanted to delete this one. I can just delete that, and then it's gone. Relatively simple, not much of technology behind it, but hopefully in a way that you can easily build these things. And again, sometimes it's really that you have to move them around to be able to see what you want to do. We will go a little bit more detailed into what you can do, but that's more or less it already. You can create your own new ones, clearly, but this will happen exactly the same way. And to clean this thing up, I can also remove it again. Good, now it's gone. Okay, Corona data set. But now moving on a little bit, and to move on, I go to a little bit of a more simple thing. This is a round robin experiment on UV-Vis, which was done by the ACE Nano project. This is only the first part. You see that I planned to include all of these 6 partners who were taking part in this round robin and then get all the data in there at one point of time. But at the moment, I just started with one to show a little bit how this can now move on and be linked to the different tools to do the management. This little sign on top of this thing here means that there is data assigned. And perhaps I should, no, I will show that later. When you click on that, you see a little bit of information, not a lot. First, you could go here, which is the more general view on that, with a little bit of a description what was done, the ACE Nano, gold nanoparticles, measurement, and there is a license, which is always important to have a license, because otherwise people cannot do anything with it. And then the main authors, Emily, who was in charge of this round robin, and then with Eva's group. Good, that's this, but more interesting is perhaps this one here. Again, you can now see here that each of these nodes with the thing has some data files associated with it. And this, again, can be quite flexible. And I just wanted to show a couple of different ways to do that, to just be able to, that you can really use a lot of different things, whatever you like to do. Okay, I first have to log in again. Going back here quickly now to this one. The first thing is here we have the materials, and we have in principle 6 nanoparticles. 5 we know the size, the 6th one we don't. one way to describe that would be, for example, in the synode as one of these inventories, the material inventory. Then you have here the basic information on these 6 materials. I have all of them have a nominal size. They have a concentration, all these things, the suppliers here and the catalog number, and therefore you also know what that size is. But we still assume that we don't. Yeah. Anyway, this is one way, just using the features of existing lab notebooks to do so. The same would work, for example, for protocols, that you say, now, what did I do here? And the first thing is I had to I got the shipment in, then I had to distribute that to the different people. Therefore, I just described that. And all that in very small things. Each of these protocols is really just a couple of steps because we have this relatively fine-grained separation from we try to build that from the instance map. Therefore, you see here, there's something missing. I don't know what the seal and the containers were, but anyway, but we say, okay, we got them from BBI solutions, and then we just made the other quads out of that and shipped them to the 5 other partners. Therefore, that's the first protocol. These ones are more interesting because here we now describe how the data was actually, or the sample was created, and then what the measurement was actually done, how it was done. But again, also here we try to keep it as small as possible. Each of these little or these parts of the experiment, we really try to break them down into these logical units, because then first the data or the data documentation can already be done. I'm waiting here for the machinery to do the experiment. I can already describe the sample preparation. Or I have my raw data, then I give that to somebody else who is doing the analysis. Therefore, I can already do everything until here from my side, then I send it over, and then the people who are doing the analysis are doing the rest. Therefore, these kind of small pieces. You can also then go in there and for the data, you can use different things. You can again use synode, the features of synode, if you like, to present these calibration data for the known particles. But you can also just use Excel or Google Sheets. If you like, if you think that this is better, then just build that in a Google Sheet, and you have the things, and you can directly link in whatever you have. At one point of time, even the curve appears. Good. Just okay. Now I cannot show that. The other thing, then I really have to jump to my one. The other thing is that in principle, we can also now create some kind of data files you have. And then this is going back to the things where I am, and these are then on my local hard drive, that you say, okay, I use these strange YAML files. And I just show you one again, because this is clearly not something you want to, or not many people want to see. But in principle, that is a way where I can really now start to structure my data and also go in the direction that I say, okay, I give some guidance on what kind of information has to be provided. I spoke already about the license, but license is important. If I show that to people and give them this file, then they know, okay, they should think about the license. They should think about the contributions, and so on and so on. And then I just have here a little bit, again, I don't want to go into much detail, but in principle, at the end, you have a lot of information. And now my cat is typing something, which is always fun. you see here that I have the basic information on these particles as well. Therefore, this is a replacement of what the lab notebook is doing. The only reason why I show that is that a lab notebook is much easier to use and much more user-friendly. The only problem, or the only thing which we are at the moment currently working on, is making the lab notebook aware of information you have to provide. These kind of metadata standards to see what actually has to be provided. Good, back to that. Last example for, or more or less, one to more little things. This is something which we are working at the moment with the University of Vienna on, but I only show that to show how easy, how simple that can actually be. Yeah, because when you now go here, this can be also just linked to a text file. Yeah, that you say, okay, I have that all a little bit similar to what I showed before with the corona study, with the cadmium study, that you just have an existing discussion documentation of these things already, and then you can link to it. Or this is an SOP you can link to, or something like that. Yeah, really making it as flexible as possible, that you say, okay, I can start already with what I have, try to think about how to restructure that, and then put that together. And at the end, we want to be more harmonized and more computer readable as well, to get the data better into something or more usable ways. But in principle, we can start with these little things and then continue with there, or with this where you are already, and then continue. I think my cat is doing something again. Good. And just really the last thing before I move over again is back to the corona data set. The other thing, what we said is, okay, these things become very, very complex. Therefore, if I would now start to put here for all these things, the data and the raw data and the process data in, these maps wouldn't be understandable anymore, and it would also not be helpful. Therefore, another point of linking to additional data is just linking to other data instance maps. That you say, okay, I have here now the size distribution. I want to go back, and then I see a very simple one again. But then I can add here all my data files, and I know I can follow the same steps without making one map too complex. Okay, that's that. Going back here. Only a couple of few last comments, and then I more or less hand over to Martin again. What you will see is that when you start doing that, and if you get the same paper and you build an instance map out of that, and then your colleague or your friend is doing the same thing, they will look very different, or the chance that they look very different is quite often. And this is an example from NanoPhase, and I think we have now 5 to 6 different versions of this one data set. I have also some 2 of the versions are in the open training area. You can have a look at them, but they really look different because you have to find ways to make them simpler. Yeah, and here in this example had the big problem that they were doing more or less the same experiments over and over again at different time points. And this made it so complex, and therefore we were thinking about, is there a way to show that better, that it's actually the same experiments, and the only change in that is really the time when they were done. And this is really a very multi-dimensional thing, yeah, because you have two different concentrations of the material and so on and so on. Yeah, therefore, it's really quite a lot of information you have to cover in there. We are thinking about extensions. We discussed already some of them. The zooming in, I said, what we really also want to do, and this is again going what Martin will present, is these controls. Yeah, these experiments you have to do, your normal lab quality control, for example, that we can cover that. We are going into much more complex test environments, yeah, really huge or very complex in vitro systems and so on. And this was an idea of these multi-dimensional experiments, yeah, that you have time, you have concentration, you have different nanoparticles in there, which you could either do all as separate instance maps, but then it's quite hard to really see the overview. Yeah, I think it's important to, when you have different nanoparticles, which you are doing the same thing with it, yeah, that you also be able then to see that in one instance map and don't have twice the same instance maps, and the only difference is the nanoparticle you're using.
We've got a question, sorry Thomas, we've got a question in the chat. Will these maps be arranged in a kind of database when shared that we can search with keywords?
Yes, this is the plan. I will go into that. Or yes, actually, this is something, thanks for reminding me, because that is something I forgot to show. Now I have to find the right one. Okay, this is the other Thomas. Yeah, there you see a lot of more things, but here I can also directly edit something. When you go here, for example, yeah, let's take the round robin. There is already an exporter here, yeah, where you can then put all these things together. It's not perfect yet, yeah, and it's also not storing the actual data files. You have to do that manually. Therefore, we are doing that, but that is actually the final idea. Yeah, that you click a button, this whole thing will put everything together and pack it into a package, which you can then put on a data repository, Zenodo, whatever, or your institutional one. But then this will then also be the way how that might get indexed. Yeah, this is the idea. We are working on it. It will take a little bit, but yes, thanks for the question. Very good. Okay. We also think about other ways to where this might end up. Yeah, and the one thing is that it doesn't have to be only experimental work. Yeah, and this MODA charter are things which are developed by the European Materials Modeling Council and the European Material Characterization Council, quite EMMC and EMCC, quite horrible abbreviation to remember what they are standing for. But that is what they came up with, a data format, also data reporting format, which when you look at that, I would say this looks like an instance map. Yeah, therefore, in principle, you have there the same things. The only main difference is, or that what we didn't discuss until now, is really this kind of setting up, you're describing the experiment, yeah, the physics behind that, and so on. Yeah, and this is where MODA really tries to get into that. It's not only data and not only protocols and data, but even going back that there are the basics of the method description. And I think that is also something which I would actually very much like to have for the experiment. And if that is just because you have that already in the guidance document, yeah, in the standard testing guidance or something like that, yeah. Anyway, you can link it to that. There is the information. Link it to it, and then people don't have to check. They know what you actually were doing. Yeah. Only again to show that it's very similar. Yeah, they then have this combination of experiments that the one is filling into the other. Again, I think this is very much looking like an instance map. And we also try to now structure the data a little bit more to show what these metadata files are. Yeah, but one important thing to remember also then when I hand over to Martin is really this: there are these defined areas which they ask people to provide, and I think that is also something we should think when we think about SOPs. Okay. The other point which we were just looking at was that you can also do that for industrial production sites. You know, this is something which I just found. We are working with a partner on that, but I couldn't show this example. Therefore, I just took something from the internet. Doesn't really matter what it is, but in principle, you can now take this industrial process and build exactly the same instance map to it, and then add things. Yeah, add here that this is at 20 degrees. Therefore, you would add here another kind of measurement or data field where you say, okay, this is 20 degrees or whatever the actual temperature was, and then perhaps probably also the color and before and after the reaction. These kind of things could all be mentioned in the instance map. Finally, we were thinking of, can we just use that also to guide people more? Yeah, and we were looking at here REACH example, where you have all these information requirements, and you can just say, okay, if I now start, if I'm a small SME and it's my first time I heard about REACH, I can go there and say, oh yeah, sure, oh my God, yeah, shape, size, yeah, surface area, blah. I have, oh my God, but I have still to look at whatever, reactive functionalization and treatment, something like that. Yeah, and you could, this is now only for the FISCAM characterization, but you can also now try to do that in that way, and perhaps even give the information that, okay, what is your yearly tonnage? One ton. Okay, then all these things, don't forget about them. They are not relevant for you. Yeah, just to show a little bit where we are thinking about where that might work, and in principle, to not make that too complicated, we would then also just link here to smaller instance maps. Okay, that's it. Now I hand over to Martin to look a little bit more into what I already expressed, SOPs. Can we use SOPs, or can we use this tool to develop SOPs?
So, hello again. This is Martin Himmli from University of Salzburg. And Thomas has mentioned my name a lot now, that we are going to show an application. And in fact, it's going to be done by Benjamin Punz, who is sitting next to me, who is going to show you a kind of a real life application. And we are now customers of this tool. So we started to use Instant Map, I would say almost recently, so a couple of weeks and months ago, and we're involved in this kind of co-creational co-development, where we did still contribute to a couple of optimizations and to kind of harmonizations in terms of what these colors mean here. And this is a real life project where we are investigating at the human hazard side, I would say, bio-nano interactions, looking a lot at the molecular initiating event, what happens to when proteins get in contact with nanomaterials. And since we are since long time involved in research that is caring about allergies or respiratory health and diseases, we use this as a model system, since we have all sorts of tools in-house for allergens, to also look at their structural alterations upon interaction with nanomaterials, certainly, and what happens at the nanomaterial, the bio-nano interface, let's say. So I hope you're not sick yet of these colors, and I will hand over to Ben, basically, who kind of gives you now an overview of what this project tries to do, what we are interested in, what the endpoints, the biological endpoints are, what we do at the characterization side of the nanomaterials per se, and then we look from there, from this instance map, into an SOP, so a standard operating procedure, which we have developed, and we also put this onto the table for further discussion. And you may want to contribute to that, and Cora, maybe to mention a couple of things that you would feel also required, call it a standardized operating procedure. Good. I guess let's have a look at what this project is.
Okay, so basically what you're looking at is the project we just recently finished. So this looks quite complex and complicated in the beginning, but what we tried to do was use nanomaterials, silica nanoparticles to be specific, and we tried to attach different chemical functional groups to the surface and then couple it, like make a protein corona in combination with an allergen called BETV1. And then we further analyzed it and tried to find structural alterations, which I will get more into a little bit later in detail. So in the beginning, as a starting material, as Thomas already said, with the different color coding, we tried to implement here and utilize it. So we had a chemical, like here, material called TIOS, and with a method in purple, we basically first synthesized the particles, and then with different further materials and chemicals, we functionalized the particles with the chemical functional groups, with an amino surface group and with a carboxy surface group. And then later on, after the particles have been functionalized and synthesized, we determined certain characterization data, for example, with NTA or TEM, FTIR, to basically see how big the particles are, if the functionalization has an influence. And also then later, in combination with our model allergen, the BETV1, we also looked at all those different kinds of stuff to see what influence it has. But to go now into what we will be talking about, into the SOPs later on, after you coupled the model protein, the BETV1, with the particles, you have to check if there have been some structural alterations of the proteins of the BETV1. And here the RBL, the Hurbel assay, comes into play. And as Thomas already showed, you can add some certain data to this node, which we did here. For example, here you can see a data file. This is the SOP, which relates to the RBL assay. And furthermore, we added a video showing how this assay is done and what use it has. And I will hand over to Martin now again.
Good. So the RBL tries to, in addition to some other characterization of the bio-nanomaterial, which is now this conjugate of the silica nanoparticles, the different functionalized ones, with the protein itself. So for quantifying, for example, how much has bound and stuff like that. So in addition, we want to have an eye on the structural alterations that might happen. And therefore, we are using this, this RBL assay. So if you go to this, then this link, and you open this, then you would come to this standard operating procedure, which we have been generating, I don't know, years back, I would say. And this is just how they look in our institution. So there is a little bit of a letterhead, and it says what it is. It's a kind of Hurbel, so a humanized RBL degranulation assay, because in dependence on the epitopes that are either accumulating or altered, basically there are impacts on the strength of this signal, basically. So reporter cells like these RBL cells would start degranulating, kind of secreting mediator molecules, like in a typical allergic reaction, which you might know. And this is addressed by this assay. RBL stands for red basophil leukemia cells. It's a cell line, and this cell line has been, as we can see here, transfected with a humanized or with a human epsilon receptor. So this is the receptor for IgE molecules. So we can use sera from patients that we get. So let's say we can do this assay, for example, for 20 different sera, so addressing the immune response, allergic response of 20 different donors, patients, basically. Yeah. And we load this onto this, we kind of interact the sera and the conjugates, and then bring them in contact with this human Fc epsilon receptor loaded RBL cells. And if these sera if there are sera specific for these epitopes, respective epitopes, then the Fc epsilon receptors are somehow dimerized, and then the entire system gives a response. Basically, the RBL cells degranulate, and there is a nice picture here. So this is the figure here. These are the basophilic cells here, and this humanized Fc epsilon receptor is shown here, and they are loaded with sera with IgE from sera of patients, and if they find their epitope on the allergen, then there is dimerization of this receptor complex, and this leads to the degranulation. And together with many other molecules, there is a beta hexosaminidase enzyme secreted, and then we can address this in the kind of enzymatic assay, which gives a fluorescence in the end. A number of things, as you can imagine, for all these type of bioassays are needed to be kind of taken care of, that have an impact on the response here. So, for example, what is an issue in immune responses is, for example, that sera may be toxic to cells. Yeah, so you need to, in addition, in parallel, run a viability assay on these cell types. You need to also take care that eventually there is complement in some of these sera, for example. So what we do is, for example, a pre-incubation with another cell type, which is this AG8 cells, for example, yeah, to kind of inhibit this complement system which might be there and which might have an impact to kind of lead to a false negative result. So a number of things need to be included into this SOP here, and it could become a very long one. What general an SOP should have, and probably you're familiar with the general establishment of SOPs, of course, it has to contain everything that is relevant, that has an impact on the readout system. It clarifies responsibilities, kind of gives an overview with really exact numbers on the materials that are required and need to be used. Eventually also on equipments that are required for that. Then gives also medical information, so safety information for the materials here, what you need to prepare. And while we're discussing in this kind of co-development phase with the other people, a couple of other more points came up, which we meanwhile have also integrated into this preparation phase, as they are quite relevant. I would like maybe to highlight just one while I'm scrolling. There, I mean, lots of materials, of course. Then here, basically the principle is explained, what I have basically explained to you, with a little bit more of details, how the system, how this enzymatic assay is, for example, run. And here we are arriving now at the kind of scheme of the assay. A relevant parameter is, of course, for example, are the cells, is this biological organism suitable? So suitability assessment needs to be done in any of these bioassays. Also here, of course, so you need to kind of, for example, be able to qualify whether the cells are fine. You need to use them in a certain passage number. This is usually also given in SOPs. We do that also in a certain range of passage numbers for these cells. We check them, how they look, the morphology checks. So there is some information on that. Also as well for this kind of cell line, which is used for just inhibiting or kind of inactivating the complement, which might be activated in certain of these, some certain sera that you address here. And then you go into the protocol, basically a kind of a nice detailed description of the protocol. And I'm not going to go into detail of that. It's taking more than 1 day, this protocol as a whole. But as a pre-requirement, I'm scrolling now along and I do not find this. For example, one thing which is very essential is that you, for example, use FCS, so fetal calf serum, which has been inactivated, because immune cells would then be activated if you do not perform this heat inactivation step. This is something that is relevant for this SOP. However, it is also relevant for other similar types of assays that involve immune cells in this instance map experimental workflow, what we have seen before. So this kind of part, how we kind of have to pre-treat this fetal calf serum or this cell culture medium would be a module that we would then be able not to have just included into this protocol here, where we have the link in the SOP or in this instant map earlier, but also we could kind of put this as a general little protocol also for other cell types, because it's a more general kind of preparation requirement. Another thing which I would like to highlight here is a very important one, and this is the interference of nanoparticles. And this is also a general module which needs to be taken care of and would be an integral part of such an SOP if you have a very long one. But since we can now couple this with instant maps, we could, for example, then also have a little piece of that. So this kind of interference testing as a general feature and as a general protocol also for other types of cells. I have said earlier that we need to do viability assessment of these cell types, which is a typical MTT or MTS or some type of assay like that. This is also a requirement that you do, for example, eventually different other steps within such an experimental workflow. So, for example, these kind of modules we can then now take from such an integral SOP, and by means of such instance mapping, locate elsewhere, wherever necessary, apart from that. Good. Another question that came up during this kind of co-developmental stage is, okay, this is the SOP, but how to evaluate this assay? Yeah. So what we then further integrated here, and here we have got 3 points where we kind of modified this SOP, is how to evaluate this assay. And with this, I guess I need to switch back to the instance map. So here to this RBL assay. And we have now linked another source to this RBL assay, which in our case, this was convenient because we had a couple of, then maybe a year ago or something, been glad to publish the entire RBL assay in the Journal of Visualized Experiments. And so we are able to just give a link to a publication of a Jovi article. And basically, here we can, what is it now? Look into, for example, how to evaluate that. And we are not going to watch the entire video, but I would just like to kind of show this to you shortly.
The bell-shaped curve obtained in the beta-hexosaminidase activity assay, indicating the monovalent occupation of antigen epitopes of immunoglobulin E due to the excess of allergen, which inhibits the allergen immunoglobulin E cross-linking at high antigen concentrations. The antigen concentration necessary for the half-maximum mediator release was calculated using the linear regression analysis. The cell viability assay was performed to exclude the cytotoxic effects derived from either the sensitizing serum or the antigen used for stimulation. 5 different human serums were used for the beta-hexosaminidase activity assay, and 4 out of 5 sera, derived from the birch pollen allergic patients, responded to BET-B1 stimulation, demonstrating that BET-B1 is a potent allergen responsible for immunoglobulin E-mediated allergic symptoms. The cross-reactivity of immunoglobulin E to homologous allergens was assessed.
So I guess with this, I stop showing you the evaluation. But I mean, these are certain aspects that need to be taken aware or taken into account. And So this concerns this RBL assay, and we have seen as also in this evaluation, there was some viability assessment required, and we know that at certain different other biological assays, like with this MO disease, monocyte-derived dendritic cell assays that I guess Ben will say a few words on. Next is definitely a similar issue. So a couple of requirements that we have to use for both of these SOPs, or for both of these experimental methods that are involved in this entire workflow. We have a certain similar requirements like heat inactivation of FCS. So we need a protocol somewhere linked as a module to several of these steps here to these protocols, or for example, viability testing, nanoparticle assay interference. I will come back to this a little bit later.
So basically, to finish up the instance map where we left off, is the combination of our particle system with in vitro studies. So as Martin already said, we used monocyte-derived dendritic cells, so-called MODCs, and we got this from blood donors extracted from PBMCs, so-called particles later on. And like there are a lot of protocols involved in turning MODCs, PMBCs, the monocytes into dendritic cells. So a lot of SOPs can later on be added here. And down the line, we basically combined the monocyte-derived dendritic cells with our particles and estimated what effect it has. This was the purpose of the project, basically. And for this, we looked at both the co-stimulatory molecule and cytokine expression, as well as the uptake, like how many particles or basically the amount of particles being taken up into the dendritic cells. And for this, we also had different protocols as well as data evaluation attached, which explains why we have so many new colors here, which haven't been previously used. But yeah, for the cytokine and chemokine evaluation, we used ELISA and a multiplex, a 45 Procatoplex. And other than that, mostly this is done with the flow cytometry, and the raw data analysis is done with some programs on the computer, which also with all the metadata files and gating strategies, for example, which will also be able to be added to this instance map tool. Yeah.
Good. Okay, so in order to wrap things a little bit up, going back to some slide format. We have shown you how standard operating procedures may be linked into the instance map, maybe kind of cut into several pieces, into modules that would be required and could be implemented at different positions into your workflow as being of relevance for some of these assays, and this somehow is now kind of maybe a little bit of food for thought for a kind of a co-developmental phase of us, where we kind of create, Thomas has mentioned MODAs and CHADAs, and the idea is actually to also create something like a biological organism data template, basically, that is relevant, that is kind of reporting on the requirements of our biological organisms. And these might be, of course, not just MODCs or RBLs or epithelial cells. This might be, of course, then also kind of environmental organisms that we talk about, kind of to have a harmonized template that kind of takes care that such type of quality assurance determining information requirements are fulfilled. Coming back to what has been mentioned, to summarize a bit, I have mentioned, for example, nanoparticle interference, and this basic information is actually derived from SOPs that we have been generating within the 7th framework program in a NanoValid research project, which maybe some people of you have been even involved in. At that time, it's long ago. These SOPs that were generated, for example, did already take care of including interference testing with nanoparticles. And one important issue is photometric readout interference of nanoparticles with photometric readouts, because they might have an influence on the photometric at the absorbance, or at the maybe they also do scattering or something like that, or even show a kind of a color or something like that. So it could be either creating false positive as well as false negative results. And there is a little bit on information on how what you shall do with it, even with this kind of dosing plate shown here in this essay. The other important essay when we talk about how nanoparticles might interfere is they might bind proteins. We are all well aware of the effect of protein corona formation, of course. So they might also bind to your protein of interest. So for example, if we look at an ELISA or multiplex system, as Ben was showing, they could bind cytokines, chemokines that are secreted, or maybe have an impact on the beta-hexosaminidase, this enzyme which we were addressing in the RBL essay. So you need to take care of that. And there is a little bit, a few words on what you want to take care of, what you want to control in order not to kind of create false negative results. Because you have just kind of inhibited the protein, the enzymatic activity for whatever. This also holds true definitely for apoptosis assay, for example, caspase activity, or any type of enzymatic activity might be impacted by nanoparticles. In addition to the other things. So there might be, this was, as I said, originally the perspective from our side, from human hazard assessment, being very much interested in immune reactions and using such type of tools. But I mean, your workflows might involve other problematic areas. And so to kind of initiate an interactive co-developmental process, how we would then kind of define such type of biological organism data templates that would be able to report impacts that are relevant for this type of assays. Here again, a couple of things that might be of relevance for cell lines, for example, just a kind of couple of ideas. If you, for example, consider respiratory impact as respiratory hazard assessment, lung epithelial cells might be something that you want to use. And this is a graphical abstract of a paper that we have published a while ago, where air liquid interface cultured cells were addressed, and there it's relevant to report in a preparatory phase about the T values, maybe show pictures of tight junction formation. And this is actually what we usually do in our papers, but it's an element of a standard operating procedure, of course, and it should be an element of such instant maps. You need to somehow characterize your air liquid interface culture. Maybe you have an eye on surfactant expression. This has an impact on the susceptibility of your cells. So food for thought, I would say, how to go on with in this harmonization process of biological organism data. I would say get in touch with us, and we have this regular instant mapping events. Get in touch with us and cooperate with us, and that we kind of come up with a kind of a community with a suggestion for biological, so BODAs, basically biological organism data templates. Good, Thomas.
Okay, then I take over. Thanks a lot, Martin, again. Just to directly follow up with what you just said, I jump back into one of the slides. This is again when I'm I hope that that you understood that this is something we plan to do, or we started to do. These BODAs was more or less an idea based on what we were working on, MODAs and CHADAs, and I really hope that from what Martin presented, it's clear what actually is needed, or that there are these things which are normally in SOPs. We are not really changing a lot. It's more that we present that in a way that these are perhaps easier to compare and to understand. That you really say, okay, you have the two assays which are the same, and you have two SOPs, different SOPs for something which is meant to do the same. Are they actually different or not? Yeah, and therefore I see there again this need for this structuring and putting that into pieces. And I think Martin already mentioned quite a lot of these, which could be these building blocks you have. Yeah, but that is in principle the first step, that when you want to do something like that, yeah, you would have to or you would really look at the existing SOPs and try to figure out what you actually would need to build them. Yeah, and when you then go back to the instance map tool itself, I think we should also think about what kind of or then these areas would be put in. Yeah, and what I already saw as a starting point is clearly we need some kind of controls. Yeah, when you look at that, and if you want to become more and more detailed in these things, then we probably would need some nodes for controls, for control handling, for quality control. We probably would have here protocols which are then exactly describing more these kind of cell model controls, which are outside of the main flow of this whole thing, but in principle these side branches which Ben already had very nicely in his instance map. Yeah, but we could also think about if we support that better by really having specific nodes for that. Yeah, that's the way to do. And what Martin mentioned is that this instance maps meeting is doing in principle two things. On the one side, we present new features of the tool, or some ideas we had about new features or what we changed, and how that is working. But in principle, it should be also a scientific, or it can move into a scientific meeting where we really look at SOPs and try to take these examples and have some kind of these community-driven SOP development that this group, and then in principle everyone who has access to the instance maps can comment on that. Yeah, that you start with some kind of a commenting tool in these SOP developments. But I hope if you are interested, please come back to us or to Martin, and then we would organize that more to really see if that is something you actually would like to do, if you see that as something which might help. And SOPs are in principle one step to regulatory standards and standard test guidance and so on. Therefore, that could in principle also move in that direction. Yeah, that it's really used in the point when you develop the SOP for a round robin, or even the guidance document at the end. Yeah, but these are all ideas which could be supported by that. Yeah, if there is interest, we are happy to then also modify that. Yeah, that you say, okay, this is just to show an empty page, but you would now move here in things and say, okay, here you have to do these cell controls, yeah, measurements, something like that. And I cannot type at the same time, but anyway, good. Okay, wrapping up, just a couple of more slides from my side. As we already had. I think I misunderstood the questions. I know that I had a little bit more time to look at the questions. I think there were the two. The one was standardization or ontology, and at the moment this is not ontology driven. Yeah, we don't have anything in there, but we want to go there. Therefore, this is coming now with the verification, completeness, and standard part. The other point was the question about or a database about these maps, and what I answered is in principle correct. Yeah, you can get you can download these data and put, and this will include the instance maps information as well. But we also have the idea to, on the one side, we have this open sharing. Yeah, the tool will be having this open sharing idea that you can look at these instance maps and they will be shared there. The other thing we plan is that you can actually have an instance map viewer. Yeah, that you, when you get a data set with an instance map, you can just load this data file into this instance map viewer and you will see the instance map. Yeah, therefore these two directions. On the one side, we have this central place. Yeah, there is at the moment only one instance of these instance maps. Yeah, therefore everything which is existing is on there, and you can share it there publicly. I would not say that this is at the moment a fair data resource. Yeah, definitely not. But it might go there. But verification, I see more in a point to the data we actually create. Yeah, therefore, what I will talk now is not about making the instance maps tool fair or even the instance maps fair, but making the data which is covered, thereby having these guidance on what actually should be reported. What these files are, which I said can at the moment be everything, but can we use this tool also to get to something which is more helpful and more, yeah, guides you to what is actually needed to report. This is a graphic I did quite a time ago, but I think it's still valid. Yeah, what we often discuss when we look at data completeness, data standards, minimal data reporting, what is it, questionnaires or checklists, all these things. What I see there is that they are really relatively broad. Yeah, they say, yes, you have to define size if you want to characterize a nanoparticle, but it's not that they really tell you what you have to report when you report size. Yeah, and there it's going there in this direction where I would like to see here something where we have these metadata collections. Yeah, that you say, okay, I now like for example what Martin just presented. If you create that, if you use this SOP, you have to make these control experiments, and you have to report these kind of things with the control experiments. Yeah, these might be the figures, yeah, the images of the cells, but it also should mention how you exactly did this experiment. Therefore, we have this translation where we go from a relatively broad thing to something which is then relatively specific and probably also experimental specific. Yeah, that you say, okay, I have a different cell model, therefore I have then to look at very specific things. Yeah, they are not general. Yeah, when Martin, these immuno cells, you have to check there a couple of things, yeah, which are different to other cell types. Yeah, therefore these things have to become very, very specific. And I hope that we can at least start with this kind of thinking very, very specifically what data is needed to report. Yeah, and then when somebody starts with that, with this kind of an instance map, and they say, okay, I want now to make this kind of experiment, then you would get already these data templates, and specifically to an experiment. Yeah, it's a lot of work, I know, but I think by the separation in things, yeah, in smaller things, there can be also a lot be reused. If you say you have these what I didn't remember the naming. Yeah, but if you say I need this control experiment for this type of cells, yeah, then you can take that and move it over into another study, and it will be the same. Yeah, you don't have to think about new metadata standards for that, because it is the same part. Yeah, if you have control levels and so on. Yeah, therefore, if you have a standard procedure how you cultivate your cells, yeah, this can then be taken over from one experiment to the other. And in that way, having these little pieces, I hope that we can do that. Okay. This whole idea brought me to this thing of an archetype, which is something which is used in an open system for medical data. But the idea is really that you just have these things and you build these structures of combining existing other things. Yeah, that you say, okay, I have here all these red boxes are something which is already defined, and then you can build more and more complex data models out of that by just combining them together and reusing them. Yeah, and if you have an author of a publication, and if you have an author or somebody doing the experiment, this is more or less the same. Yeah, you can define a person as an archetype and then reuse them, and you don't have to always think about that again and again and again. Yeah. One of these articles, or just going back to this YAML thing, but I still hope that this is not actually so complex to understand. But all these things are just hierarchies. Yeah, therefore, this is something where you would say, I put these things together, and a property would have, in principle, a standard form, yeah, or an compound would have the standard properties. You can guide them, that you can record on the storage. You definitely have to record on the manufacturing, where all these comes from. And since we have these things already, people would hopefully not forget about it. Yeah. A much easier example is references. Yeah, it's amazing that you still think, or every journal thinks that references have to be exactly done as they want to do it, but they still have always the same information. Yeah, and this is why reference management software is working. Yeah, you agree on some kind of a template, and then you reuse that all over again, and then the computer can take this information and build an reference out of that, which looks exactly as this journal wants to have that. Yeah, but it's not any kind of magic, and you don't have to do it differently. And now I knew that I had forgotten somewhere and set, and now I know where it is. Anyway, and again, little things. Yeah, you start building from smaller things and then build them together. Similar to an instance map as well. Yeah, you start from you build these little things, and then at the end you have this very complex study. And a very simple starting point is just a quantity. Yeah, something which is a number, but a number with a unit, yeah, or which is associated to something, yeah, like size. Yeah, you need a value, you need a unit, otherwise it's not a quantity. Yeah, if you forget the unit, nobody can use it. Yeah, therefore take care that this is in there. Yeah, and if you have larger, larger than, smaller than, something like that, you can also define a modifier. You can then go to person. I said person is something which might be relevant or might be harmonized, and we can actually now, with the curate system or something similar, we can even directly get this information without anybody having to put them in over again. Yeah, because curate is taking care of that, and we get name and some other basic information. Yeah, the same is then for institutions. Yeah, but you don't have to define these over again. Yeah, and the question was on ontologies. yes, here ontologies are coming in, because I just need first name, I just need middle name, and so on, as assign that to the ontology, and I know what I'm talking about. Yeah, here it's easy. If you go to the more scientific part or the more essay specific part, then we probably have to define the ontology or the ontology terms, but in principle it's possible. Yeah. And then you can go here to the property steps, and what I just wanted to show here is that you can then build actually these things out of these smaller things. And you have here somewhere licenses, references, contributors. Yeah, this list of contributors, and this I don't have to specify that anymore because I know that it's a person, and therefore everything else is defined. Yeah, and in that way we can in principle build this archetype for a protocol. You have a license, you have contributors, you have references, you have then the steps, and in the same way you could do that for an SOP or for a whole essay in principle. Yeah, and this is how we then at the end would put all these data together, have these pieces. Every of these pieces would be something like protocol archetype or an instance archetype, and at the end everything is a complete data set. It should not look like what I just presented, because I understand that nobody wants to write these YAML files other than me, or no, there are definitely other people as well, but it's quite interesting, but it's not something we want to do. Yeah, therefore this is just the picture from Ace Nano, just to show, yes, if you know what we have to prepare, yeah, you can also do that. We have nice user interfaces, or you do it in Excel. It doesn't matter, yeah, as long as we define somewhere this way how to present that. Yeah, that's more or less first everything from my side, and because I didn't want to forget, and we have now more an open discussion and open demonstration wherever we want to go. I wanted to say thank you first. Big thanks definitely to the Nick and NanoPhase teams, because otherwise it's just I'm not smart enough to come to this kind of very simple but great idea. Thanks to all the instance map testers which we had, which really now is also working or doing exactly, telling us what is working, what is not working. People who provided the data in a way, that I can use it and also show it in a way, some way, and then clearly to Jochen Meyer and the NanoCommons Consortium for technical, or Jochen Meyer are the people who are actually doing this tool, and the NanoCommons people are also the people who design it and help me help us. I shouldn't say me all the time. Anyway, help us to create this thing and hopefully make it as useful as possible. That's it from my side. Next step would be 2 very simple questions to just get a little bit this thing starting, hopefully breaking the ice. And then the final thing would be build your own first maps, where we can guide you a little bit through the process again. It's already quite late, but I'm happy to stay and discuss that, or even show a little bit more how that actually works, that you can start with that. Yeah, but also then later, if you have questions, please just contact me and I can help you setting that up. Thanks, and then over to Christine for the Mentimeter. Now, this is just a question of what you think these ideas we had, experimental workflows, that is what we discussed most of the time. I showed the MODA workflows where we can also, as I said, apply that. The regulation data is this idea of presenting that to people, or even showing that you provided everything you wanted, you need to provide for if you want to apply something. I didn't talk about nanofabrication so much. Yeah, I talked about nano materials production. I think these are very similar, yeah, that you go into more industry-based experiments, yeah, and then bring in there also these kind of on-the-fly measurements. Yeah, this is where you go in nanomaterial production, that you have a continuous flow of the different things coming in, yeah, and really in situ, also the size measurements, for example. These kind of things, yeah, and then the other two questions are clearly different. This is where you see more definite, more use for it. Is it really this presentation to others, like Ben did really nicely today, to explain what you actually did, and then the data connection. Yeah, and what I see is that experimental workflows is clearly the favorite. Probably because we talked the most about it, yeah. And it's clearly the thing why we created that, yeah. Therefore, that's fine. 7, that is 50%. Perhaps we can get a couple of more people, but otherwise we.
Yeah, should I move it over to the?
Yeah, I think you can
next,
move it over.
yeah, people can still come back to it.
We also ask when you go out of the meeting, you will also get a very short survey. There are these 3 questions. We have now 2, and then there is a 3rd question. Would be you can answer there. Very quick introduction to this question. Yeah, you saw that we have these basic informations, and the basic informations are at the moment really just the name and a short description, which you see directly in the instance map tool. Yeah, you see licenses, which are probably not the first thing you would think of, but if you have some ideas what the tool itself should clearly at the beginning directly present, yeah, that you get some idea of what this protocol is or something like that. Yeah, if you have there some ideas, and if not, that's fine as well, or if you have to think about that, it's fine, but just having the idea of what you think might be needed, yeah, to really be quicker than having to go first to your to the data file which is behind that. Yeah, it might be something like just the final value. Yeah, when you say their size measurements, that might be that you just say, okay, this is the average size or something like that. Yeah, that you say existence of data. I think that is what this little icon is, if I can interpret that right. Metadata link, yes. Nanomaterial properties, very complex thing. We were, just to mention that, probably some of you heard about that, about our INCI thing, identifier, where we try to put a lot of data or properties into a string. It might be, I hope that this works at one point of time, that we just prepare the INCI and then you have some kind of visualization which then present you these properties in a nice way. Yeah, shell types and so on. But that is just that this is not so boring. I just continue to talk about things. Life cycle, okay, that's good. Materials, we have assays. Names of assays is probably what is meant there, which is good. Yes. Links to other maps. These are the links which are in there. Yeah, perhaps we can perhaps we can find some way that you don't have to click on properties, that this is really in the box already, something like that. We have to think about that. Okay, data connections. Yes, I think you like the idea that this has, if attachments are there, I think the idea of having that also for data clearly shows. Ontologies. This is probably not what would be visible, but we are thinking about adding ontology tools to that, that you, when the data is coming in, you can then also directly associate or find ontology terms, the corresponding agreed ontology terms to something. Metadata completeness scores, that's something which I somebody else has to find a way to do that, but yes, that's definitely would be something, or at least you would clearly show how much is already in there. Yeah, this is that you really use that as a tool for data progressing or data collection. Yeah, that you say, oh great, you end your day and you say, oh, I have, for this step, I collected 100% of all the data which I have to collect, something like that. Yeah, would probably be also nice. Okay, good. Thanks a lot, Christine. There are some little things which I could quickly show. Yeah, that I some things which might be frustrating, or perhaps not really frustrating, but perhaps not clear what they actually mean. And this is mainly here when you start putting properties in. I think the main part is quite simple. Yeah, you have these different types here. You have to be a little bit careful that you really see this cross when you start putting connections from one to the other. At the moment, you can connect everything to everything. Yeah, who knows? Somebody who might be familiar with the NIC approach, they were relatively strongly defined rules to that. We made it very flexible at the beginning, where you can really start connecting everything to everything, to just understand what is really needed. Yeah, is it clear that after each protocol there must be another instance, or would there be another protocol coming in? Yeah, or is there then what we often have is, for example, that you have two different starting materials, where the one is the biological system, more or less, yeah, and the other one is more the material itself, yeah. And then you do something with the one, then you do something with the other, and then you put them together and do something with the whole system, yeah. And then after that, this would then, yeah, I said you have to be careful that the cross is moving, yeah. And then you do this, yeah. Therefore, this is relatively often, or at least I had the problem that I needed this from time to time. Yeah, that this combination of multiple things coming together. Yeah. But other than that, there is relatively not much to say. Yeah, the removing always, yeah, if you want to remove something, you click on it. It's also, this is unfortunately not so perfect. If you want to select this edge, then you can also here with settings always remove it. But you really have to be very precise where you click to really select that. This is unfortunately a little bit about this library we are using, but there will be also updates on that. This is the one part. The other part is that you have in principle everything which you do is auto-saved. Yeah, therefore, whenever you move here something, this will be saved. You can undo it. Yeah, but if you now end your session, you will come into this setting directly. Yeah, therefore, you have an auto-save. This is not when you look at other people's maps. Yeah, then you do that only for yourself. That's and then very quickly, okay. When you go here and then add something, the only thing which is not auto-saved is when you change here something. Yeah, if you save something and then go out, this will not be changed. Yeah, you have to first save properties and then you will get that. Yeah, reasons for that I can explain, but not that. I don't think that this is quite so interesting at the moment. That's it, more or less. Yeah, you have your properties. You have always in the settings when you want to delete something. One point, yes, what I want to you can share with your group, and you can be in multiple groups. Therefore, if you really want to also use that perhaps in your group or something like that, in your in your research group, we can set that up for you. Yeah, but at the moment, the user management is still a little bit on my side. Yeah, therefore I create users, I create groups. But if you want to have one, you can have multiple. Yeah, and this is more interesting if you look at the other one, because there I'm in many groups. Yeah, if I'm here, then I can really share that now with where do I have into settings? Yeah, I can have that with my alpha training or alpha testing team, the training team, and then I already prepared another part project group where we will discuss some other things. Yeah, therefore you can be in multiple groups, you can be sharing with multiple groups, but also be careful that everyone know or sees that. Yeah, therefore be aware of are you really knowing who these people are? If you need to know who they are, I can share that, or more or less. It's personal data, therefore it's a little bit problematic. But don't share anything which is priority, confidential with people who you don't know. If you have, for example, your group, your research group, then sure, you know who the people are. The alpha testing people know who the alpha testing people are. Yeah, but for the training, the only people who knows that is Christine and me. Yeah, therefore, be careful when you choose something. I think that's it. Yeah, we can. If you have questions, please. I can stay longer. I'm happy if you want to see something. I'm also very happy to present that. Yeah, if that was too quick or if it was not clear, then I'm happy to stay here also a little bit longer. Other than that, I would say thank you. Please try it out. If you need anything else, yeah, if you need another group, if you need access to something, if you have ideas of new features, please also let me know, and then we can arrange. And if you need a personal training, I'm also happy to do that from time to time. Yeah, or join the alpha testing group, because then you get your personal training every other week. Thanks a lot.